So, hello everyone. So, in the DNA replication of prokaryotes, we have completed the initiation and elongation process of DNA replication, and now today we are going to talk about the termination process in prokaryotes. So, we know that in elongation process, the replication fork moves in both clockwise direction and counterclockwise direction, because in prokaryotic DNA replication, the DNA is already circular in nature. So, two types of replication fork moves in opposite direction. One of the replication fork moves in clockwise direction, another moves in counterclockwise direction. So, when both replication fork moves in opposite direction and a new strand is formed against both parental strand, now when will it be terminated? So, this is a major point. So, what happens that in case of prokaryotic DNA replication, the DNA contains a special type of termination sequence that is called TER sequence. TER sequence, TER stands for termination. So, at the opposite region of origin of replication in prokaryotic DNA, there is the presence of a special type of sequence that is called TER sequence. This TER sequence contains the 20 base pair sequence, and several TER sequences are present in this termination region. So, we can see here different types of TER sequences are there, like TER J sequence, TER G sequence, TER F, TER B, TER C sequence, TER A sequence, TER D sequence, TER E sequence, TER I and TER H sequence. So, now this TER region is responsible for the termination of DNA replication process. So, now another most important point is that this TER sequence alone cannot terminate the termination process. This TER sequence bind with a special type of substance that is called TUS sequence, TUS, that is called a TUS. So, TER sequence binds with a special type of substance that is called TUS. TUS means terminus utilization substance. So, when there are several TER sequences are present, but one TER binds with one TUS and form a TER-TUS complex. So, always remember, TER-TUS complex is now responsible for termination of DNA replication. So, here we can see one of the TER sequence binds with TUS and form which complex? It will form TER-TUS complex, TER-TUS complex. So, the major feature of TER-TUS complex is that when a moving replication fork enters to TER-TUS complex, it will stop, it will halt. So, suppose this replication fork moving in clockwise direction coming to this TER-TUS complex. So, when it comes to TER-TUS complex, this replication fork, this movement of replication fork halts. At the same time, when another replication fork that move in opposite direction comes near the TER-TUS complex, at which one of the replication fork is halted, then another replication fork also stops at that point. So, finally we can assume that there are two daughter DNA formed from one parental DNA, and both are attached at a single point, that is at the TER-TUS complex. So, now there is only one a step is needed that can cleave the attachment at tortoise complex, and that will finally form two daughter DNA. So, here we can see that after the formation of two daughter DNA, so all parts are separated, but both daughter DNA are connected at a very small point, and the DNA is attached in this way is called catenated DNA. Catenated, simply we can see here, suppose this is one of the daughter DNA and this is another daughter DNA. So, when both daughter DNAs are attached at a single point of attachment, then this is called catenated DNA, catenated DNA. So, now there is a need to separate both DNAs, both daughter DNA from this association. So, there is role of an enzyme that is called DNA topoisomerase 4. DNA topoisomerase 4 causes the separation of both daughter DNA from its attached point, and finally, there will be formation of daughter DNA. So, finally, we get two daughter DNA from one parental DNA. So, this is all about termination process, and finally, from one parental DNA we get two daughter DNA, and the process is terminated in this way. So, thank you very much.